Okay, so for link reaction to take place, inside the mitochondrial matrix, But one thing that we need to know here, it must be under aerobic conditions. Okay, so why must be aerobic conditions? We need to learn it also, but not now. Will be learned in the oxidative phosphorylations. Okay, so these link reactions, we have two important events actually take place. One is called decarboxylations, another one is called dehydrogenation. Decarboxylation basically means that removal of CO2. So CO2 is removed. Dehydrogenation basically means that removal of hydrogen atom. Okay, understand the concept here. So the enzyme involved here, decarboxylations, you need to know the enzyme. So the enzyme is called decarboxylase. Okay, dehydrogenations, the enzyme is called dehydrogenase. So very simple, right? Okay, so in this case, decarboxylation takes place when 3 carbon pyruvate molecules are decarboxylated, removal of CO2, and then hydrogen atom is removed from pyruvate, transferred to NAD. So it means that when NAD receive hydrogen atom, so I say that receive hydrogen atom, it become reduced. So reduce NAD. Okay, so the process is known as dehydrogenation. So what is the process now? Look at this diagram. Okay, now 3 carbon pyruvate, 1 carbon, 2 carbon, 3 carbons. Okay, 3 carbon pyruvates. So these 3 carbon pyruvate, what we're going to do? We're going to carry out decarboxylation, remove CO2. Can you see that? We remove CO2. So when we remove CO2, the remaining part is known as acetyl group or acetate. Are you clear? The name of these 2 carbon compound here, the remaining one, is termed as acetyl group or acetate. So this acetyl group or this acetate cannot enter into the next part of the aerobic respiration, which is Krebs cycle, unless we have a carrier. So this carrier is termed as coenzyme A. So this coenzyme A going to bind with the acetyl group to form acetyl-CoA. So this acetyl group is 2 carbon. Are you clear? So coenzyme A is something like a carrier to bring this acetyl group or acetate enter into the Krebs cycle later. Are you clear? Okay, so what we do now, can you see that? Decarboxylation take place. The first process, decarboxylation. Remove the carbon dioxide. And then what we do next? We remove the hydrogen atom. So in this process, we have the oxidation. So NAD will accept the hydrogen atom, become the reduced NAD. So this one actually is mean reduced NAD right now. Okay. And then, so we have decarboxylations and we remove the hydrogens, correct or not? We remove the hydrogen atom. So this part actually is number 2 is dehydrogenation. So it means that when you remove, so remember we say that when you remove H atom, so this process is called oxidation. So therefore we can see that pyruvate is oxidized. Correct or not? Okay, we remove the hydrogen atom. Okay. Now, how about NAD? NAD, now you can see that it gained hydrogen atom. Correct or not? So when NAD gain the hydrogen atom, so NAD now is reduced. So reduced to what? To reduce to become reduced NAD. It's in a reduction process. Okay. So in this concept here, some students ask, I didn't see any hydrogen atom released, right? So in fact, you have to know this one, can I see that CO2 double bond here? So the electrons was shared. So cleave here, okay, try to imagine now, we cleave here. Cannot. When you cleave here, what will happen in this direction? CO2 removed, but C have double bond O, C. So it means that C double bond O and C, when they cleave it here, so it means that we lack of 1 bond. So hydrogen atom will bind here later in the intermediate process here, which is not shown, the hydrogen atom. So then we remove the hydrogen atom, join with the NAD. So it means that then it can react with the coenzyme A. So when you react with coenzyme A, can you see that the coenzyme A now can carry these acetyl groups to form acetyl-CoA. So this acetyl group is 2 carbon. Are you clear? Okay, so decarboxylation. Okay, right now, number 1 is decarboxylation. Number 2 process is dehydrogenation. So what is the function of NAD? So NAD actually, they are coenzyme. Okay, so coenzyme we learn in enzyme topics, right? What is the function of coenzyme? They are functional group carrier. So NAD actually is hydrogen atom carrier. Okay, they are hydrogen atom carrier. So they carry the hydrogen atom for what? Later we can see it, okay, in oxidative phosphorylations. Okay, so but the process here, okay, the process here, for pyruvate to enter into the mitochondria, you must have a transport protein or carrier protein. So therefore we say that the membranes, okay, the mitochondria outer membrane, inner membranes, they do have this transport protein for pyruvates to enter into the matrix of the mitochondria. But to do this, we need to have oxygen. Are you clear? We need to have the oxygen. So it means that it must be under aerobic conditions. Okay. So what are the products formed here? Okay, so understand, per glucose molecule, we were going to have 2 molecules of pyruvates, correct or not? So therefore, 2 molecules of CO2 is released, 2 molecules of reduced NAD will be formed, and we need 2 molecules of coenzyme A to form 2 molecules of acetyl coenzyme A. Okay. So list the product of link reaction per glucose molecule. So per glucose molecule, we're going to have 2 molecules of carbon dioxide, okay, 2 molecules of reduced NAD, and 2 molecules of acetyl coenzyme A. So I put 2 carbon because 2 carbon refer to acetyl group, because coenzyme A have more carbons atoms. Are you clear? Clear? So I summarize it later. So first we have pyruvate. So this pyruvate is 3 carbon. We total have 2 molecules of them came out from glycolysis. So where are them? They are in cytoplasm. So means that this pyruvate have to be brought into, okay, into this mitochondrial matrix. For link reaction to take place. Okay, so what the pyruvates do? Pyruvates actually undergo few processes. First, we have the decarboxylation, CO2. So 1 pyruvate remove 1 CO2, so total we have 2 CO2. Okay, and the process also involves the oxidation and reductions. So we have the NAD, 2 molecule NAD, receive the hydrogen atom, so to form 2 molecule of reduced NAD. Okay, so in the process, we do need a carrier. So who is the carrier? Coenzyme A is the carrier. So coenzyme A actually will take this acetyl group to form the acetyl-CoA. So acetyl group is the one, the 2 carbon, coenzyme A, acetyl-CoA. So this acetyl-CoA later will enter into Krebs cycle. Are you clear? Okay, so if I label, so what is the first process? So the first, this one confused. So I just write down, so decarboxylation take place. Okay, we remove 2 molecules of CO2 as the waste product. And here we have the oxidations. Pyruvate is oxidized. Reduce NAD. NAD now experience reductions. So during oxidation, what will happen? Dehydrogenation. Okay, dehydrogenation actually take place. Okay, so this is the whole process of the link reaction.